Ben, we haven't met, I think.
Yeah, that's right. So nice to meet you. I'm
Too.
working with Mike.
Oh, okay, very good.
So Ben, maybe just to give 30 seconds about your background and so on, so
Yeah,
that
sure. Well, by training, I'm a physicist, a computational physicist. So I started working with evolutionary algorithms and how to use them to find ground state structures in molecular systems. And this turned out to work well, but not as good as I wanted to. So I started looking into a little bit more sophisticated things like reinforcement learning and so on, try to give every molecule which tries to find its place kind of a brain to self-organize. And I think that's kind of the overlap I have with Mike, so to treat self-organization kind of in a gentle way.
Hmm.
And yeah, we are now working on a few projects which relate evolution and morphogenesis. So we had a recent paper where we tried to answer the question whether evolution can go faster if it's working with competent parts or with cells which can do things. So if it's faster if these functional parts of the gene are manipulated rather than a structural part of the gene, for instance, if you go for direct phenotypic encoding. So the answer is yes, it can. Not always. It's dependent on noise, so it's a little bit complicated. But depending on the system parameters, so if you have kind of realistic noise in the system, evolution can really well utilize these functional genes. So to speak in machine learning language, we use this neurocellular automata for morphogenesis experiments. And in this case, so if you have noise in the system and a program which is not a problem which is not super simple, then evolution can very efficiently utilize its competencies for self-organization. And on top of this, it's also very efficient in transferring to different problems, for instance. So if you manage to get these cells to grow a particular structure, it's very simple or very efficient to transform the goal into a similar structure if you have these functional genes available. So if you go for more direct phenotypical traits, then this is much more complicated for the evolutionary process to find.
So Ben, by functional gene there, do you mean genes that regulate other genes?
Well, in this neurocellular automata, you have a neural network in every cell, which kind of is the update or simulates the update process of this cellular automata.
Mm-hmm.
So that's the functional genes, the parameters of this neural network, which regulate their own states.
Okay.
In this case, so the cell type.
Cool. Yeah, it's so interesting, the importance of noise. This has been a theme and something I've been, in all of the sort of developmental stuff that I've been working on. But also, you just see it in so many parts of neural signaling. And it seems to me there's just some very, very fundamental principles at play in relation to noise and variability and how organisms or systems can take advantage of it. In fact, it's totally a feature, not a bug.
Yeah.
In fact, that they can tune it, they can release it, more or less of it when they need it. And yeah, I was actually writing a paper kind of with a number of people about variability and noise as a part of the system, as an ability that organisms have and can take advantage of. I must get back to it because there were too many authors on it. We were trying to pull it in from many different angles. And yeah, everybody's schedules just it was too many. So, but must get back to it because it's absolutely fundamental. And I think not well appreciated.
Absolutely. Also in this system, we kind of found a functional relationship between how competent or what's the optimal level of competency of these cells and the level of noise, so to say. So there's always a trade-off in how competent the cell wants to go, if under quotes, of course, if we leave the competency as a parameter to the evolutionary search, so as a gene.
Yeah.
So if you're very low in noise, then you can go direct. But if you are very high in noise, then you need more competency to solve the problem.
Yeah.
But leaving the noise as a parameter would also be interesting, actually, in our system.
Yeah, the trade-off between stability, and evolvability is there, right? It's the explore trade-off, and yeah, it relates really in terms of things like just developmental robustness as itself a genetic trait, which can manifest as a tendency or, you know, possibility or risk of ending up channeling into states that we call pathogenic, pathological, which I think, you know, the genetics is just reinforcing that. The genetics of things like schizophrenia and epilepsy and autism and so on, it's all pointing towards the idea of just general developmental robustness as the underlying risk phenotype. And the phenotypes that emerge are actually nothing to do with the molecules underneath. They're just properties of the system. They're emergent attractor states in the system. So it's a much more sort of generic underlying risk that's all about noise and how much the system can buffer it.
I mean, another way to think about noise too, consistent with that, I think, is that by committing to the fact that you have fundamentally an unreliable substrate where, you know, unlike the way we encapsulate it in computer science, where, you know, the higher levels can depend on the metal being good and all that. In this case, if you know that everything is going to change, so your low level details are going to change, you're going to be mutated, you can't never ever know really how many copies of anything you have, then that forces the kind of architecture that you guys and we have been looking at, where you don't take the past literally, you sort of compress it, but then there's the creative forward end of that funnel, that bow tie, where you don't really know exactly what happened, and it doesn't matter because you know it's going to change anyway, and you have to, you know, it facilitates making these kinds of systems which just have to do the best they can in the current circumstances, which may mean interpreting the old stuff in different ways, just because you could never, you know, guarantee. And I sort of tend to, I think of kind of a spectrum, and I don't know to what extent these examples are real, or we just haven't found out enough, but you can kind of think about maybe C. elegans being on the low end of that spectrum, where it's pretty stereotyped and it just kind of assumes things are going to be, you know, be what they are. And then you move forward from that into mammals and amphibians, and then planaria on the other end, which is like, you know, they'll figure out a way to do the right thing no matter what you do to them, almost. You know, we can think of it like that control knob of how confabulatory you are and versus.
Yeah, no, I thought I really enjoyed the way that you had phrased that in that recent paper that you sent, not the one today, but the previous one. About the planaria. Just the fact that you basically can't make mutants of them that have any phenotypes. I didn't know that, actually. That's super, super interesting. And so the idea that they're at this far end of this spectrum, where, you know, they need their genome because they have to encode proteins and stuff like that, but actually the regulatory web is like just maintained on the fly, not so much by constantly referring back to the genome saying, hey, what should I be doing? What are my settings? So it doesn't save the settings in the genome in as rigid a way. I just found that super, super fascinating, but also sort of explains why planaria may be unusual in relying on things like electric fields so much, you know, maybe more than ones that do refer back to the genome more often.
Right. So we have no evidence yet that the C. elegans care about the bioelectrics, you know, really at all. In between, you know, amphibians and mammals, we have pretty good evidence for a lot of stuff. But planaria, I always thought it was interesting that in planaria there are no mutant lines except for the two-headed and the cryptic lines that we made, and ours aren't genetic. They're precisely the bioelectric ones, right? It's wild that that's so. I think one thing that is interesting about bioelectricity in particular, although I'm sure it's probably true of biochemistry and biomechanics too, is that it's a really convenient mechanism for tying together the kind of neural connection, for the kind of neural connectionism that you guys were talking about with actual development. Like, that's literally the same mechanisms. You know, I think evolution really just loved that symmetry, and, you know, and really made use of it. So this is something that you know, has drew from the very beginning when I started to work on bioelectricity. Like, that's what I wanted to get to, is to see if there was a symmetry between how it's used in brains and why it's such a convenient feature, right, for cognition, and could we use that to explain this like amazing problem solving capacity of embryos, what you were just talking about, the robustness and the
and, you know, one of the other upshots of having noisy molecular components is that relying on single mappings of one gene to one phenotypic thing becomes just a bad strategy, right?
Yep.
You just can't do that. And instead, this sort of logic of fields and landscapes and collective global constraints, to me, is just a much better picture of how developmental biology works, whether it's bioelectric stuff or molecular fields or some combination of interaction between those. Which is, it goes against, I think, the trends in developmental biology over the last few decades, which has been find the molecule that does the thing, find the signal, where's the receptor, where's the pathway, what's the transcription factor that turns on the things, and then there we've solved it. And then, of course, it's a nice, tidy story, and people love those until someone comes along and goes, oh, yeah, but you know what? That same signal somewhere else with a different receptor, or even the same one, does something completely different. Context is key. And it's just interesting to me in terms of the sort of the sociology of the science of that field, that there's been this absolute drive for nice, simple, linear, reductive stories. And I think people have realized we've exhausted that paradigm, and we need to go back to, and I really mean go back to, because the older way of thinking about fields and landscapes and so on.
Yeah, but you know what else I think is interesting is that as far like I think that's true, but I think there's this also massive resistance especially among kind of the more organicist thinkers in developmental biology against computational metaphors. So like, you can get flack for bringing in any kind of connectionist cognitive thoughts, but you can also get flack for, oh man, you're saying it's a computer, it can't be a Turing machine, and like people really don't like some of the computational stuff, which I think is a shame because it has such utility in certain places.
And it's funny, I don't know, Nick, if you've gotten any flack on that front yet. I'm expecting some. I've gotten a little bit. I'm certainly expecting some, because we're making this proposal, and I know, Mike, you've made a similar one, that the genome is basically a generative model. That's what it encodes, which is, in the sense, it's a compressed representation in a space of latent variables that allows the developing organism to construct a new instance of the type, which is whatever the species is. And in the scheme that we have, that's sort of analogous to this variational autoencoder, which is a machine learning thing that learns to categorize, say, pictures of horses or something like that, and it learns abstract horsiness through these compression through multiple layers. So it abstracts the sort of invariant features of horsiness, and then it can decode them through an expansion of layers in the decoder to produce a new instance. So for our analogy, evolution is doing the encoding to get this compressed representation, and then development does the decoding. And that to me feels pretty apt. And the idea of the genome as a generative model just sounds like a description of what the job description of the genome is. But the analogy with the variational autoencoder, I think some people are going to say, oh, look, here they are. They're just reaching for the latest technology, and it happens to be AI, and it's just a fad. And it's like when we thought brains worked like clocks or hydraulics or whatever the latest thing was. But I do want to say that for me at least, there's a distinction here with the idea that the genome is a program and it works algorithmically, because variational autoencoders and other models like that don't work algorithmic. They're trained algorithmic, but then the model is not an algorithm, right? The model is not a program. It's a set of, it's embodied as a set of weights. And so we have these new things, these large language models and other things, which are new kinds of things in the world that we don't even know what they are. Like, we have to have a natural science of them. Yeah. It's not like we just program them and and you can say, here's this bit of the program does that, and this bit of the program does this, and it happens in series like a nice clean algorithm. So I'm hoping that people don't take it that we're making a strictly sort of algorithmic claim, when we're making that comparison, because it's not like that at all, because those things are not algorithmic.
Yeah.
I don't know, Nick, what you think of that, if you've had any feedback so
Yeah,
far.
Received, I think, kind of similarly very mild but poignant comments in that direction. And yeah, I tend to agree that I'm and I'm even less interested in the analogy directly of, oh, the genome is generative. That, yeah, the lining up of these two things, I think, is fairly straightforward, and many people have made this in the past, but it's, if we use this as a model, as a substrate, how can we poke at the computational thing and ask questions that give us insight into the biological thing?
Yeah.
Yeah, I mean, I think
that's the goal.
Yeah, I think one thing that happens sometimes is that, especially kind of on that more organicist end, is that people are pretty willing to accept that just because you know the parts of a biological system, you don't really know the capabilities of the whole, right? That people are pretty willing to accept that. But I think people really still think that if you have something that is either literally an algorithm or something that can be simulated by an algorithm, then they know everything it does, and that does not capture this magic of biology. And this is one reason why I love these very minimal computational systems. And we recently did something with sorting algorithms. We don't even really know what bubble sort actually does, because if you look at it the right way, you can see some amazing things that it's doing that are not explicitly in the algorithm at all. And so I think if we extend to the same humility that we like to extend to biology, to say that just because you know what the parts are, you don't really know what it's capable of until you study it. It's, as you said, it's a new thing in the world. You have to empirically study it. I think if we could extend that to the computation and not assume that we know everything that it does, then I think we could be okay. But yeah,
I think it lends itself equally well. You could say you could build it as a computer system, right? Of course, these neural networks are built in silicon, but they effectively model collective dynamical systems that generate energy landscapes that force the configuration of the thing into sets of either local or transient minima if there's a trajectory. So it's not the perspective that you take on it is a choice in that sense.
That's right. That's right.
And for me, it's the same as the choice of looking at your embryo and saying, you know what, because BMP7 is here and it binds to this receptor and turns this on, it's a really linear reductive mechanistic view that isolates it away from the context of every other molecule and so on. That's a perspective that's not wrong. It's just an incomplete perspective, because when you change the context, when you don't control the other variables, when you put it in another part of the embryo, other things will happen, and that's important to know as well. But it is funny, people do have prejudices, and you know what, there are some philosophers of biology out there now who are really, really pushing against what they call gene centrism, as you know, Mike. And to me, they're overdoing it. Partly they will say, look, genes can't specify traits in this one-to-one way that is claimed. And to me, that's kind of a straw man, because I don't know many geneticists who make that claim. But it's still, they look at it as, they look at the evidence for genetic effects on a one-to-one basis, one-by-one basis, and say sometimes there's evidence for affecting something, but other things do as well, and it's all complicated. And sometimes they get to the point of saying, by rejecting gene centrism, they reject the function of the genome as a whole somehow, which is just a weird move and unnecessary, right? I mean, you can accept that genes don't do everything, and they don't drive things. They're not actively causing things, while also accepting that cats have kittens and dogs have puppies because they have cat DNA and dog DNA. So accepting that genetic information is a real thing, just that it doesn't have a simple relationship with the ultimate phenotypes that emerge. So I'm hoping that the model that we present can marry those things, right? And can kind of reconcile, because it's not genetically deterministic,
right,
one-to-one linear simplistic mapping, but it's also not a free-for-all, like anything can happen, and sort of in the middle, I hope.
Yeah, I mean, one thing about that, the straw man is that, I mean, I think you're right. I think explicitly most people would not at this point push that view. However, it has implications in, for example, biomedical approaches. So I do think that most people think that, oh, CRISPR, genomic editing, we're going to fix all this stuff because, and I'm like, okay, there are some single gene diseases, but after that, what are you CRISPRing after that? And I think, right, and I agree that this model has. And models like it have a good chance to provide that layer of actually, okay, so questions of causation is one thing, but then now practically, right? So how are we going to use these to actually make the changes that we want? You know, how do we drive it?
Yeah, and again, for the kind of work that Nick does, and I guess Ben, that you do similar, the idea of in silico models that we can use as sandboxes to try and figure these things out. This kind of generative model encoding is I like to think of it as sort of 2 steps indirect. It's not just that it's not specifically encoding elements of the outcome. It's not even individually encoding or directly encoding parts, separate processes. It's collectively in this weird compressed way, leading to those processes being constrained in such a way that they produce this thing. And to me, that's a powerful encoding. It's really versatile. It's very evolvable. But it's, but I also think, I've been thinking about how to go about actually encoding it in the kinds of systems that you guys have. And it's a challenge, and partly because what you also need is just the self-organizing processes themselves. They just need to be there. Those are the things that the genome is constraining. Yep. And so in your systems, you're going to have to encode some physics of the stuff that you're making your bodies out of. And I know, Nick, that your soft-bodied robots kind of have that physics in there to a certain degree.
Yeah, to a very limited extent. I think this is a really nice connection with Mike's work, is that we think about, because there's such an indirect relationship between the things that are encoded and the final outcomes, that there must be some really complicated process of going from one to the other. But because this is a complex adaptive system and that behavior is emergent, that you can get really complex, chaotic behavior with very, very simple rules as long as you iterate them over time. And, thinking of the, layers of the decoder as, steps in a developmental process, that, what's actually happening within each layer of a neural network, going from some set of features to some other more or less abstract set of features, is a very trivial transformation. And yet doing that a bunch of times gives you these weird, crazy things that we no longer understand. And Mike, the way that you're pointing out the pieces of and information pathways in which biological development does that too, and showing how we can manipulate very simply a limited number of pathways, and yet doing that within an actual process that's unfolding over time gives you amazingly interesting and weird phenotypes. I think that our current soft robot systems don't have that much complexity and unfolding, and that we've bought into a system that kind of purposefully ties up a lot of that to make it simpler to compute. But the idea of that process and the regularities and generative nature of them being the magic sauce here, I think is really interesting, and something I hope we capture a little bit in our current systems.
Yeah, I mean, it's all the stuff that the genome doesn't have to encode, and that's partly what gets it away from this overreaching claim of the genome just directs everything. It doesn't have to, right?
It doesn't have to.
But mostly it just doesn't have to.
Yeah.
We have proteins that have properties. When they're expressed in cells, they'll interact with membranes to cause physical tension, to cause things like a tube to form. or a vesicle to bud off, or other morphogenetic processes, and the genome can constrain what happens by directing the expression of different sets of proteins with different properties in different physical domains of a developing embryo. And I think it would be really interesting to try and set up a model system like that that's quite simple. Say something like, I shouldn't say quite simple, but relatively simple, rather than making a whole thing, but like patterning a hand, right, a limb.
Yep.
And so there's some morphological processes that happen, this condensation of like cartilage cells is sort of like a simple Turing diffusion process. There's a few things like that where the physics are fairly straightforward. You've got a developing field. It can develop like this. It can develop like a whale's flipper or a bat swing or whatever. And it might be a nice little test system where you could say, okay, I'm gonna evolve the underlying genome in different ways, or allow it to evolve in different ways, and see if it can find constraint dynamics that produce interesting differences in the phenotype, in ways that you could even apply some selective pressure to, if it needs to swim, or if it needs to grasp, or if it needs to fly, or whatever. Because that's one where the physics is constrained enough, maybe, that you have enough to go on.
Yeah, that's a really interesting. I like the limb also because there's lots of stuff that's worked out, as you just said, but there's also puzzles like why you have 2 bones in your arm, not known at all, despite the tons of molecular data. Like, why are there 2 instead of 1 or 3? Nobody knows. And so I like that a lot. We do, so partly Ben's work and some other stuff. We have a bioelectricity simulator that is, more or less biorealistic, and we do that and gene regulatory networks. And the idea is exactly as you said, to see if like one thing that we've been working on is the electric face in the early frog development. So the thing that maps out the eyes, the mouth, like where all that stuff is going to be. So to see, what does it take to evolve something like that? And Ben has some nice work on it. Actually, another thing about GRNs, which goes to the point that Nick just made about the emergent complexity, and also to Kevin, what you were saying before about different perspectives and attractors and all of that. So like all of those obviously are there, but there's another perspective which gives you some yet additional capabilities, which is that when we started looking at biological GRNs, and this is mostly like real biological ones, random ones don't do this as much. It turns out that if you look at them through the lens of simple behavioral control, so just training experiments, right? So we found 6 different learning capacities there. So habituation, sensitization, so Pavlovian conditioning, and you can get to that, but once you're willing to test the system in that way, right? And that also has biomedical implications. So I think this idea that there are multiple perspectives, and each one sort of facilitates certain things and doesn't particularly enable other things is valuable.
Yeah, and I think the question of that's a good illustration of what do you get from applying the perspective of machine learning, connectionist network kind of ideas to gene regulatory networks and so on. And Yuri Alon has really nice work on this. He wrote a great book on systems biology, and I've been teaching some of this to our undergrads, because when I first encountered his way of thinking about things, which is very much an engineering physics kind of way of thinking about things. Why are the circuits designed like this, right? You have 2 genes that turn each other off and they turn themselves on. Why is that? And that, what that gives you is a bistable switch, right? One or the other. And that's a simple, obvious one, but it's where the engineering physics perspective is just super useful, right? And you can see why in a developing organism or like a lambda phage that's deciding whether to be lytic or lysogenic, that's a good thing to have a nice tight control. But you can look at multiple kinds of levels where those systems, regulatory systems, get more complex. They've got extra layers and so on. And one of the really interesting things is that you can get effectively multi-layer perceptrons in out of those systems, which do pattern completion. Right? So even if the signals coming in are a bit noisy and they're not the full range of all the signals that you would expect, the system basically just does pattern completion. It says, okay, I've got enough of these, and then the interactions at this level fill out the rest of it, and it can execute the developmental program for a limb or a heart or whatever is appropriate. So yeah, it's a really powerful way of moving, I guess, of seeing the logic of what the system is designed to do, while still allowing that it's made out of messy biological components. In fact, it's because it's made out of messy biological components that it's designed this way. Not just, one of the really interesting things points that Yuri makes really well is that when you look at the types of motifs that you see in gene regulatory networks, you only see a subset of the ones that could exist. That's because those are functional and useful. But even of those ones that are functional, you see a smaller subset. Those are the ones that do the functions robustly. And that's really key. And it's a super important message that again, the biological systems are just dealing with noisy stuff, and therefore they must land on the motifs that work that way. Really powerful insight.
Yeah. Andreas Wagner has some amazing stuff on this. Have you seen some of this? It's really good. So
Andreas, one of his main points is that because these systems have to be robust to noise, as a consequence, they become robust to genetic variation.
Right.
And that allows genetic variation to accumulate, which is, can be cryptic in the sense that it's not affecting anything because of the robustness until it reaches a certain point, or it's put together in a certain combination, or environmental conditions change, and then you can get a new phenotype. So yeah, it all ties together. The robustness and the evolvability are completely linked with each other.
Could I want to get your guys' thoughts on something. Let me just share a screen for 1 second. I want to show you. I'm sure you've seen this before, but I want to see what you guys think of this. Do you remember this stuff? This is Darcy Thompson's deformations from growth and form, right? So he puts these species on the grid, and then you do some sort of mathematical deformation to the grid, and you get something else that's that's a real animal. So and there's in the book, there's all sorts of skulls and bones and everything else. Do you think that so what's this grid, right? I mean, this has been driving me crazy for years. Is like, what is the actual grid? It's clearly something, but what is it? And how does that map onto the actual developmental biology? And I wonder if I'll stop this now. I wonder if the kind of thing that we're doing now with these encoders and so on, like, do we now have, because of that, do you think, what do you guys think this is? Do we have more insight into that?
I don't know, Nick, if you have any thoughts. I mean, for me, what springs to mind is that we can think of these gene regulatory networks as shaping these energy landscapes in the sense of the landscape there being patterns of gene expression that the cells could adopt, right? So that's like an internal landscape. But you can also think of it as being physically distributed across an actual physical landscape, right? The actual physical stuff of the embryo. And I've been thinking about this in terms of axon guidance and the growth of nerves, how nerves are guided. And there, it's like there's a molecular landscape that they follow, but they're also following a physical landscape. They're sticking to other nerves. They're the rigidity of the axons is part of the force that pulls them one way or another, and so on. So I think you can have kind of levels, right, where you've got energy landscapes at a molecular level within a cell that are effectively descriptors of attractors in a dynamical system space. And then at a higher level, as you distribute different cell types across a domain of actual cells, then you're going to get what's effectively an energy landscape. Gradients of protein expression. It may be gradients of ion channels that could create electrical gradients as well. I mean, what's interesting about them is they regulate. That if you truncate it, they'll form the same gradient over a smaller distance. So there's collective self-organizing capacities within there. That's what is really interesting. But I mean, Mike, I'm sure that none of what I just said is any news to you or very insightful. But that's the idea that you're nesting the same type of process at a molecular level within cells, and then at a cellular level across fields of a developing embryo. To me, it's a helpful way of thinking about it, even if it's vague.
Yeah, no, definitely. I wonder what you guys think about do we have like I showed this to a student years ago, and she said that's great. Where's the knob? I want to twist the I want to stretch the grid with, like, for real in a real embryo, right? So so do you guys think that this computational formalism helps us to do that, to be able to use the kind of interventions that are available to us to actually deform the grid as a whole without saying, okay, so this cell has to go here and that cell has to go there? Like, how do we get access to these large scale features?
Yeah, I don't think it helps. Well, I'm not the person to go and tell you what the knob is in biology, but I think this formalism of thinking of it as a generative model, absolutely, go to your classic L system of grow a tree, and yeah, there's one little hyperparameter in that system that tells you this is how long each branch should be, or this is the angle in which you see branching happening. And yeah, you see exactly the same sort of thing, you see in Darcy Thompson's book, of the same pattern but stretched or mutated and deformed in some way. And we're not formalizing, we have the language and here's the knob within that to change, but taking that conceptual framework would suggest there is a knob somewhere. If we're trying to get the analogy of that in the particular example of a neural network, then yeah, we can do some fancy tricks to try and interpret what's happening within each neuron or synapse of that network to say, oh, here's a knob that might be good to tweak and do walks through the latent space, or perturb the decoder in some specific way that I think, yeah, would get you things that do in fact shape very much the way that happens. And I think that's also an interesting question of kind of what the ideal use case of this analogy is. Is it, focusing on the evolutionary part of how does this infrastructure come about such that we have this regularity, or to say, hey, look, a lot of these creatures that we see in biology share these same regularities. Do we just have to find them once, and then we have this enormous palette to work with, the way biology does, where so much of what we see is constrained according to one very good set of rule sets versus doing the search over the rule sets themselves.
Yeah, I mean, the one thing that's this is something that Daniel Lobo and I talked about some years back, is in particular for models like L systems, right? These feed forward emergent models. What's frustrating about them from the biomedical angle is that absolutely there are parameters that will change things, but going backwards and saying, okay, but I want to change like this, right? So for morphoengineering, for regenerative medicine, you want to be able to say, all right, but I want this. What parameter do I change, right? And for those kind of emergent systems, that's really hard. And I think biology does have an answer to it, and so I'd like to take advantage of that. So, for example, here's a crazy idea. I don't know what you think of this on the computational side. Like, remember Morvintsev's deep dream stuff, where you push it? Okay, hold on. That's interesting. I don't know what that was. That was weird. Yeah, weird. Yeah, well, you push it backwards through you push the pattern backwards through, and you ask, okay, what is your ideal represent? So I wonder if we had these models properly parameterized, if we could do something like that and extract the archetypes, for example, of some of the things that we see in morphospace, and then you use that to say, so what kind of prompt do you need to be able to shift it in basically control of large scale features? I want control over large scale features, not of molecular features.
So, Mike, what's interesting about this question, right, is when you say, is there a knob that changes this thing? The question is, like, is it a single knob in terms of one molecule, right? Is there one molecule that controls whether your fish is long like this or wide like that? And it wouldn't surprise me if there were developmental mutants that did affect that kind of form in a dramatic way, where there's like one Wnt protein or something like one BMP, whatever it is, that has a dramatic phenotype. And I think a lot of Evo Devo stuff is also looking for those nice clean stories again, where it says the difference between this fish and this fish, what happened in evolution was this gene got mutated and they went from this boink all of a sudden to that, right? To me, that just is just super, super unlikely to be the way that evolution happens. That suddenly a fish is born that's like twice the size of all the other members of its species, and it's the founder of a new, like, that's just not the way it is, right? So what's interesting about the encoding, the way we're talking about it, is that it's this distributed collective encoding. Which then the question is, well, how do you get evolvability out of that at all? If it's so distributed, if each of the latent variables in there is affecting so many things, and each of the traits is affected by so many variables, which is what we know from human genetics and quantitative genetics, is all the traits are really polygenic, and all of the variants are really pleiotropic. They affect many different traits. It seems like you should have gridlock. And what's really interesting in the analogy with the artificial systems is that they have the same kind of problem with the way that they do the encoding in connectionless network, but they have these orthogonal disentangled representations that emerge when there's some kind of pressure to make them that way. Or when there's some connection costs as well. And, you know, there's other kinds of factors that lead to this emergent modularity, where it's not a unigenic modularity, it's polygenes, but they're overlapping, but they're in separate dimensions of the representational space, right? This abstract representational space. And what that means is that to me, is a great explanation for why it's actually possible to select for, either artificially or by natural selection, particular traits that you can get to move really far, right? You can, like, artificial animal breeding and is incredibly potent, and plant as well, without, you know, affecting too many other traits until you get to really kind of extreme sets. And so to me, that's one of the things that got me personally super excited by working through this model, is that we weren't thinking about that going into it. That popped out of the comparison with the machine learning stuff and these disentangled representations. And it just seems to be a really apt way to think about the genetics of complex traits, where, you know, what it means is that to get at them, maybe the best way to get at them is to select for them and then see what got selected. You know, in an interpretability sort of use case, is like, do the experiment. Don't go in and don't say what can I tweak. Say what am I selecting, and then ask what got tweaked.
Yeah, that's very interesting. And I mean, yes, I completely agree. When I say control knob, I don't mean one gene or one protein, but there are some convenient. So, like, for example, I'll give you a couple examples. One is there's a particular bioelectrical state that will induce regeneration of tails at a tail location, but limbs in an adult frog. And so it's very context sensitive. We sure as hell don't know how to micromanage either of those things. We didn't have to say which stem cells, which anything, it's sort of, so it has this feature, like, okay, it's not a single gene for sure, but it does have this sort of bow tie node feature that all the stuff afterwards is handled. It's coherent. It makes sense. It's sort of integrated together. So I'm hopeful, just on the biomedical side, I'm hopeful to identify some of these, some of these control knobs. And then other stuff that we play with has to do back to the kind of cognitive analogies, which is that if we take from behavioral neuroscience reagents that they have for modulating attention, modulating precision, modulating information processing, and things like that, then you can do interesting things like apply SSRIs to regeneration, or hallucinogens, which we're doing now, or serotonergic modulators, which we've done for years. And you can chase, for example, you can chase a planarian with a very particular type of head into a different species shape head with with that species brain shape, with that species distribution of stem cells, and so on. No genetic change needed, and it's a very simple. I mean, I don't know what controls the parameters of the head shape, but you can get them to do that, and you can get them to you can get frog tails that are shaped like zebrafish tails, or like a different species of frog face and things like that. And you can change a precision. For example, one of the things in a more recent paper that's under review right now is talking about how if you use certain kinds of cognitive modulators that reduce anxiety and precision, then basically you still get developmental outcomes, but they're stochastic, and it's as if you've taken off the pressure to get it exactly right. It's sort of like, yeah, we'll make this head, that head. It's whatever. They'll make something, but it's not. They're just not as that error parameter. That's like no, it has to be within a tolerance. And otherwise we'll adjust until it is. That's just kind of taken off. Yeah. So I want to see this push like together with some of the other formalisms that are trying to merge things that are happening in neuroscience with developmental biology. To see how much of that we can basically steal, and reappropriate, because I have a feeling that what we're onto here, I think, is a very deep symmetry, between the formation of cognitive systems and the formation of bodies. I think there's a deep symmetry there, and I bet we can reuse a lot of that stuff.
Yeah, I mean, the analogy, thinking of development as a cognitive problem that the organism has to solve and the cells within it has to solve, makes sense to me. I know some people will object to the use of cognition in those terms, but I think it's perfectly defensible if you just define cognition in terms of using information to solve problems under potentially novel scenarios to produce adaptive outcomes. Then
exactly.
That's a type of cognition. The kind of cogitation that we do in humans is another type of cognition. Yeah. And we don't have to inflate one or deflate the other to draw analogies between them. I think that's perfectly valid. What's interesting, I mean, you got me thinking about, what I said earlier about what the genome doesn't have to encode, right? It doesn't have to encode basic physics. It doesn't have to encode the fact that, negative and positive charges attract each other. You can take a lot of that as read. And it's interesting in an evolutionary sense, when we're thinking of pushing phenotypes one way or another, that in many cases, the bits that you're tweaking also don't have to encode all of the outcome, because there's other bits that will do the work. So this bit that you're tweaking doesn't have to tell them how to do it. They know how to do it. It just has to stop them doing one thing and impose a different constraint regime so that the possibility space is narrowed over here instead of over there. And so what you get, I think, in that sense, is this sort of hierarchical relationship of constraints. And to me, that's really similar and analogous to the hierarchical relationship of constraints in decision making.
Yes.
Where I can choose. I mean, even like in prefrontal cortex, I can choose a goal that acts as a top-down constraint on parts of motor areas that pick out activities that I should be doing to achieve that goal, which themselves act as constraints on the primary motor area, which is selecting actions to do right now in the service of that activity, in the service of that longer term goal. And I think that idea of hierarchical constraints, which in that case are operating over nested timescales. The prefrontal cortex goals are over a very long timescale. And then within that, they're giving some top-down information that constrains stuff on shorter timescales and shorter. It seems to me like you can draw some similar analogies in developing systems. And in fact, you can think of the gene expression system as reconfiguring things on a fairly long time scale relative to the biochemistry that's doing signal transduction, for example. And when you think about that way, you get this interesting kind of segregation of what's going on at each level, because what's going on at the level above it is too slow to show up here, and what's going on below is too fast. And you get these sort of independent domains. So you can get some modularity that emerges that way, because you can tweak one level and get effects at the lower levels for free, because that's the architecture of the system.
Yeah, no, that's fantastic, and I think we have an opportunity here. So we've been calling that kind of thing a multi-scale competency architecture, where each layer deforms the action space for the layer below, exactly as you said, so that it doesn't have to, there's autonomy, and so you don't have to micromanage all that stuff. So I think we have a really good opportunity here. I've really wanted to do this in a much more kind of quantitative way, is to actually look at the information passing through the different layers in this kind of architecture, and really kind of more rigorously tie it both to developmental processes and to the decision making. So I love that aspect of it. At the attention, the modularity, and all of that, the goal setting. I think we should do that. I think we should try to make a model where we can trace that happening, and I can almost visualize like the main figure from this, where you've got the network in the middle, and then we sort of show the cognitive case and the developmental case below, and just show exactly the mappings of what is happening layer to layer to enable the autonomy, the delegation properties, all that kind of stuff.
Yeah.
And the timescales. The timescales are really critical. We haven't done much on that at all.
The timescales really give you a causal insulation, and they allow for some causal integrity at a single level. And then sort of top-down effects and so on. Fernando Rosas?
Yeah, I do. Yeah.
Fernando has a really nice paper out recently with a bunch of other people looking at this kind of macroscopic causation and insulation between levels from a computational point of view. And I should read it again, because it's actually really nicely demonstrating, first of all, the reality of macroscopic causation. It's just not the case that all the causation comes from the bottom up. And this is something that, we've been dealing with in discussions around free will, because there's a notion that everything is deterministic down at the low level, and it leaves no room for any other causes. Like we've exhausted all the causes deterministically at the level of atoms and molecules. Everything else just emerges bottom up. And in fact, you can show that that's demonstrably not the case. It's even just not the case in very simple physical systems. But in more complex systems, it's not the case because they've evolved for it to not be the case. Right. So once you add selection in there, then you get macroscopic causation towards a purpose and a function. Then you get systems that naturally have this scale, a functional scale, where at different things that are happening at different rates and in different parts of the system really have an independence from each other. Not complete, of course, because they're talking to each other. They're just not, they're not one causal web.
Yeah.
They're segregated causal webs.
Yeah.
That feels like a really powerful method. And, I agree. I get this same sense, this intuition of something really powerful there. And that the idea that if you could get a system where you formalize it and where you could play with it, that would be just a really powerful experimental theoretical system to have available. So
and I think these questions of the different time steps and evolving towards them is an interesting one. That you have the various timescales across development, but thinking about the generation of these things over longer evolutionary timescales, it's interesting about questions about how these things arise when the selection pressure for them is many generations down the road for things like robustness and adaptability, and I could see cases like Wagner's work, where you have very reduced selection pressure that lets you build up enough mutations to have some complicated thing down the road. But I think the more interesting versions are when there's indirect pressure for that for whatever other reason, that modularity in the brain is extremely helpful for a number of reasons, but presumably comes around just because of physical space limitations, connections, costs, energy constraints, where it's like the here and now gives you this longer term property too.
Exactly. And that, I think that's absolutely right. And a lot of the longer term things that come out are due to very different immediate pressures, like the evolvability coming out of having to buffer noise, which is just there right now. Like evolution can't select for future good evolvability, right? So those are really good examples where the sort of immediacies that are at play inform the way that the system can evolve, the spaces, the possibility spaces that emerge. I was going to say something else and I forgot. Shoot, what was it? It'll come back to me maybe. It was another example within there of the sort of the immediate concerns that the system has to deal with, but nope, it's gone.
I'll hop in and say one thing quickly then, which is I forget exactly how you phrased it, Mike, but that the Planaria's constraints are relaxed under the hallucinogen that allows it to find this new phenotype. That it's relaxing some of them, but there's also very much of the canalization and error correction still happening, that it doesn't create some totally random thing, but a well structured phenotype of a different type of organism. And maybe I was misinterpreting that, but I think it's interesting to pull that to your question of, in this computational model, how do you think of what things change what? And it draws me to like the deep neural networks are easily fooled idea that, you know, we can go in and perturb a few pixels and this thing that looks like a dog, it'll be extremely confident it's a cat, or vice versa. And usually we're interested in those at the end of the process in the phenotype themselves, because that's what the classifiers are looking at and will give us these interesting counterintuitive outcomes. But thinking about that at the earlier stages within your latent space of where's the opportunities where you're, you know, on some ridge in the manifold and can just kind of knock things into another part of the space. I think, you know, absolutely we could use those same tools to move around in that space in order to give you whatever frog face you want instead of the traditional stereotyped one.
Yeah.
But yeah, I'll have to think more about that too.
Cool. Okay, thanks very much, guys. This was super interesting. Lots more to talk about. So
yeah,
we'll be in touch.
I remembered what I wanted to say. It actually was about the credit assignment thing in evolution, which is a really interesting analogy there, in that there's just a very different solution to the credit assignment problem in machine learning with the backpropagation algorithm than there is in evolution, where it's this highly parallelized search across a population. Right? I mean, it doesn't do it in series by training. It does it in parallel, and then it does it again and again. There's an interesting analogy there. And then the other, I mean, Nick, in relation to what you were just saying, the idea of these atavistic phenotypes that we sometimes see. This, you know, when you have a mutation, you mutate the system, and you get this throwback to what looks like a more primitive, evolutionary primitive thing, where there's like a memory of it, a latent memory of a phenotype that evolutionarily these things used to make, and then you can reveal that. That to me, that's a super interesting thing, and it fits really well with the latent variable compressed generative model kind of analogy, and it doesn't fit well with the blueprint program sort of metaphors at all, actually.